A group of proteins that have recently changed our thinking about protein structure and function are the so-called metamorphic proteins. These proteins are capable of forming more than one stable folded state starting with a single amino acid sequence. Although it is true that multiple folded conformations are not ruled out by the laws of physics and chemistry, metamorphic proteins are a relatively new discovery. It was known that prion proteins were capable of folding into alternative structures, but metamorphic proteins appear to be able to go back and forth between two stable structures. While in some cases the metamorphic proteins undergo this switch in response to binding another molecule, some proteins that can accomplish this transition on their own. An interesting example is the signaling molecule lymphotactin. Lymphotactin has two biological functions that are carried by its two conformers. A monomeric form that binds the lymphotactin receptor and a dimeric form that binds heparin. Lymphotactin is a chemokine that recruits T lymphocytes and natural killer cells and is produced mainly by activated cytotoxic T lymphocytes and activated natural killer cells. The function of a protein is determined by its shape. The shape of a protein is determined by its primary structure, which is the sequence of amino acids. The sequence of amino acids in a protein is determined by the sequence of nucleotides in the gene encoding it. The function of a protein, except when it is used as an energy source, is absolutely dependent on its three-dimensional structure. Each protein has its own unique shape. If the temperature or pH of a protein's environment is changed, or if it is exposed to chemicals, these interactions may be disrupted, causing the protein to lose its three-dimensional structure and turn back into an unstructured string of amino acids. When a protein loses its higher order structure, but not its primary sequence, it is said to be denatured. Denatured proteins are usually non-functional. For some proteins, denaturation can be reversed. Since the primary structure of the polypeptide is still intact as the amino acids haven't split up, it may be able to refold into its functional form if it is returned to its normal environment. Other times, denaturation is permanent. One example of irreversible protein denaturation is when an egg is fried. The albumin protein in the liquid egg white becomes opaque and solid as it is denatured by the heat of the stove and will not return to its original form. Not all proteins are able to refold, and how proteins normally fold in a cell appears to be more complicated. Many proteins don't fold by themselves, but instead get assistance from chaperone proteins, the chaperones. Although the three-dimensional structure of a protein is determined by its primary structure, it may need assistance in achieving its final shape. As a polypeptide is being synthesized, it emerges from the ribosome and the folding process begins. However, the emerging polypeptide finds itself surrounded by the watery cytosol and many other proteins. As hydrophobic amino acids appear, they must find other hydrophobic amino acids to associate with. Ideally, this should be their own, but there is the danger that they could associate with nearby proteins instead, leading to aggregation and a failure to form the proper tertiary structure. To avoid this problem, the cells of all organisms contain molecular chaperones that stabilize newly formed polypeptides while they fold into their proper structure. The chaperones use the energy of ATP to do this work. Each protein has its own unique sequence of amino acids, and the interaction between these amino acids create a specific shape. This shape determines the protein's function, from digesting protein in the stomach to carrying oxygen in the blood. There are a number of agents that can disrupt protein structure and as a result denature the protein. Changes in pH and salt concentration alter electrostatic interactions between charged amino acids. Changes in temperature, especially higher temperatures, reduce the strength of hydrogen bonds. Presence of reducing agents break sulfur to sulfur bonds between cysteine amino acids. If the protein is subject to changes in temperature, pH, or exposure to chemicals, the internal interactions between the protein's amino acids can be altered, which in turn may alter the shape of the protein. For example, pepsin, the enzyme that breaks down protein in the stomach, it only operates at a very low pH. At higher pH, pepsin's conformation, the way its polypeptide chain is folded in 3 dimensions, begins to change. The stomach maintains a very low pH to ensure that pepsin continues to digest protein and does not denature. Let us see how this dynamic works in the case of enzymes. Because almost all biochemical reactions require enzymes, and because almost all enzymes only work in the best way within relatively narrow temperature and pH ranges, many mechanisms regulate appropriate temperatures and pH so that the enzymes can maintain the shape of their active site. None of the agents, such as heat, salinity, and pH, breaks peptide bonds, so the primary structure of a protein remains undamaged. When a protein is denatured, secondary and tertiary structures are altered, but the peptide bonds of the primary structure between the amino acids are left intact. Since all structural levels of protein determine its function, the protein can no longer perform its function once it has been denatured. Thanks for watching.